So yeah, this program is structured in a way where it addresses people who are beginner in coding, or who wants to cover bioinformatics, or who wants to cover data science for uh, biomedical data practically through logic. And one step, if you completely understand and the logics behind using these different methods on the data, then you can expand your knowledge to either using one of the coding platform R, or using an alternative coding platform Python. So all the associated resources that I'm going to show you today is given in the program page. So let's quickly go through the introduction to machine learning in biomedical data, which is in course 7. So this course as such will give you a very brief introduction, and you will learn about various different machine learning methods of analysis, including supervised and unsupervised machine learning methods, as Elia talked about, and how they apply to different biomedical data. And you can also see examples of multi-omics integration and applying different data science techniques to those integrated multi-omics data to answer the biological question in hand. So let me go through the first lesson in Introduction to Machine Learning in Biomedical Data with you, so that we can get a sense of how the lessons are organized and what to expect from these lessons, and how you are going to maximize the learning potential of this platform. So here we get a very brief background about machine learning, and why we are going to use machine learning, and why machine learning methods are successful in getting applied to different biomedical data, and why they are being taken and why they are being considered as the next level of analysis for the sheer volume of biological data that is available. And then you go through the next session, which is a purpose of this course. In this course, this tells you what this course actually teaches you. This course will teach you about the methods. This course will teach you about the logic behind the methods, and this course will make you understand how that logic is applied to different kind of biomedical applications. And in the process, you will also in the process of doing what this course recommends you to do, you will develop critical thinking in how to approach a biological question in terms of data science methods. And most of these data sets are curated data sets that are derived or that are taken from the public resources, from already published data. So you will understand what it means to extract, or what it means to extract additional information from a data that is primarily recorded or primarily registered to answer a question in mind by the authors. And going through this experience, you will also learn how to, as Elia actually mentioned it, in some of the examples, some of the project examples, you will learn how to specifically answer a question that is related to a particular data set in hand, and how to tailor the method, all the methods, different methods, and the logic that you learned to suit to the data that we have, that we consider in hand. So we preach and we actually practice learn by doing method. So there is no waiting time in terms of learning what you are going to do. You do this analysis, and you learn as you go along in this project work. So the next section actually talks about how does machine learning work? What is the basic behind? What's the logic behind? How does this machine learning work, and what's the logic behind these algorithms? And as Elia mentioned, there are two major classification of these machine learning algorithms. One is unsupervised and supervised. I'm not going to go into detail about what is unsupervised and what is supervised, but on the broad scale, if you have different groups in your data and you don't know about the segregation of those groups, then you can use unsupervised machine learning, and you can employ different algorithm or different set of algorithm to arrive at a result that will segregate your data into multitude of these groups. And Similarly, in a supervised machine learning, you know something about the categorization of your source data, and then you can learn, or you can label those data based on the groupings, and you can set a training data set and a test data set for your machine learning algorithm to learn the pattern hidden in the data that defines these categories, and then try to apply that to a new hidden test data and to categorize that data into one of the available labels. So again, here also, we have a multitude of different training data sets, multitude of different training algorithms and that you will employ, and that you will learn the logic behind these algorithms in a very intuitive way in the big data bioinformatics platform, the Talbot Bioinfo platform. So when you go through this, then you go towards the applications of this machine learning data, where there is humongous potential for these machine learning methods to be applied to various data sets, like in clinical data sets, in pharma, biotech. Those are only two. I mean, like in agricultural data set, in the main future lies in integrating all these different, not only omics data, but also different types of data from different sources, like the patient demographic information, like the patient history information, like the history of disease in the patient's genealogy information. All these data will come together in a very integrated way, so that the machine learning algorithm can learn and understand the pattern behind those data to be applied to the various applications that we say. So we also give links to some of the projects where you will find a specific question is answered using a very detailed methodologies, and you can go through, or you can follow those steps and understand how the approach is designed, how the method is designed to answer those specific questions. And there are also other reviews and literature information given. So all these lessons you see here that are logic based conclude with the conclusion and a quiz, which I and we all recommend you to take, so that you can evaluate yourself how you have processed the information that you had been going through. So if you are not satisfied with the results, you can go through them again and then learn it and process it much more detailed, and attempt these questions. And these quizzes are there to expand your own version, your own understanding of where you stand. So that completes lesson 1 in the machine learning introduction to machine learning biomedical data. So if this lesson and this particular course, as I mentioned, addresses the basic, the main logic underlying the machine learning platform. Now, if you have mastered that main logic underlying the machine learning analysis of biomedical data, then you want to expand your knowledge in applying that logic using a different an additional platform or additional environment, let's say like in R or using Python. So let's go through how such an analysis is dealt with in R. So this course is Introduction to Data Science using R course, where we start from the very basic. So one of the very main challenges in trying to get exposed or trying to learn a new platform or a new environment is to how to load data and how to get familiarized with the different information and different environment that is present, different aspects of this environment from this coding language. So for R, which is a freely available language, and there is tremendous amount of information that is available. So we start from absolute basic, how to load your data. So you have your data, and you have a download, or let's say you have downloaded the curated data set that we present you for your analysis on the Bioinfo platform. So you want to do the same analysis using R code. So we start with really very, very basic, like how to install R on your computer. So once you go through this, how to install R on your computer, then we go through the codes that is necessary to load the data that you have in hand into the environment, into our environment. So for this purpose, we have the code blocks here that tells about how this is done, and comments inside the code blocks that tells about what this specific line of code performs, and a lot of information in and around the codes that explains the syntax and that explain different parts of the code that so that you understand and you get familiarized with the with using R as a platform to load your data. So this is just one example where you load your data. Excuse me. I'm sorry. Yeah. So where you load your data using read.table function. It's one of the function that is present in R codes, and then it takes some of the other inputs, like if to let the coding environment know whether or not your data has a header, that is the column names present in the data, or whether or not, and how the fields are separated in your data, whether they are tab separated, or they are separated by comma, or separated by any other special character. So we mentioned this, and then we go through different ways or different source of files that can be loaded into R. For example, here you can use OpenXLS library to load Excel file into R. And why do we need to do that? We just we want to expand our knowledge into R, which is a very powerful platform, especially for machine learning analysis and for data science methods, where many of the other easy to use and GUI method like Excel are really lacking, and we do not have Excel to way to deal data with such a huge volume. And then, as I mentioned, we can also find code that will explain you how to load data that is comma separated. So, it's not only about loading data. Then after loading data, we want to have a peek, or we want to have a visual feedback of how the data looks like. So we can perform different functional analysis, like we can learn the dimension of the data that tells you about size of the data in terms of rows and columns, and we can print first few lines of the data, that is the head function, and also we can use the print function to print the entire data frame or data in your hand. And while you are going through these lessons, you will get these snippets of information that will educate you on the necessary technical terms and technical terminologies and technical aspects of R that is absolutely necessary for the analysis, for your downstream analysis. Like we are going to deal with a lot of matrices, and we are going to deal with a lot of data frames. So what is the main differences between matrix and data frame? That is also given here. So after you have gone through these code blocks and the syntax explanation of these codes and different methods to achieve the same target, so we have a section called Try It Yourself, where we give you a code block. This is a console on itself where it can run our code, and then we give you small little exercises where we take the examples from this above section, and then give you in an incomplete syntax form, so that you really understand which part of the syntax is missing and how can I modify this code and how can I make this work. So here your main idea is to import and view a text file. So you can use the text file link here and substitute where this actually goes. This is where the path of the text file goes. I will just copy it, copy the link address, and then I will select this portion where I have to replace in the code block to make this code work, and then I will paste it. So that completes this, but I mean, to my view, to my eyes, it looks complete. Let me run it and see. And it says there is an error running code, and it actually tells you what the error is. Okay, so I go through the code one more time and see that the syntax is missing. The necessary part of the syntax is missing. So that is, this is a table that I want to read. So I complete the syntax and clear the console and try it one more time. And again, it says it is successful in running the code, but the answer is incorrect. Okay, that gives me more information. And why is that so? If I see through this, this particular data that we have printed out here has the column names written on top of this. And I have not instructed R to consider that my data has a column name embedded into that. So I do that, say that header is true, the option for header is true, and let's clear it and run it one more time. And bingo, we got a successful code. We got our code running successfully, and the answer is also correct. So this is just one example where you can iteratively expand your knowledge in understanding how our codes work. So this is a very basic example of reading in a table and printing out what the table actually gives. So this such an example is given for all the steps right throughout this course, for even the complex steps that involves machine learning methods and analysis. So this is extension of the same code for considering Open XLS platform, and this is for the CSV file. And in addition to this, we also have some extra biological data set where you can try the above code using the different forms of data sets. So if I want to try this one, I can copy the link of this code and put it as substitute in any one of this or the appropriate console, and then try the output here. Or if you have followed how to install R in your own system, then you can copy this code and then paste it on your own R console, and then try to see how you can execute this in your own computer. So you can get a sense of understanding how R deals with the different variety of data. So this is, yeah, so such a method or such a approach is extended to how to read data from outside of R, let's say from your command mode input, or from from the command line input, or from a terminal. And then we get an introduction to what is an R function and what is a user-defined function, and how we can employ that in our code so that you don't have to write the same set of codes again and again and again and again. So after you complete all these sections, so you get to practice your own block of code, where we give you an example set. Again, you can run this and see the answer is correct. So this is a simple function that calculates the number of characters in the string that we have given. So I just can verify if it works for some more, and then it will work. So yeah, so that is how this one works. So you go through and complete the lessons, go back, and you go to the next lesson, which involves or which talks about data processing and visualization. That will be our next session. So when we now I want to show you how a similar lesson works in the Python environment. So R and Python, we can employ both of them for machine learning analysis in this course. It's about your preference to select which one of them, but there are basic differences in how these are and in which environment these are employed. Like R can be installed in your computer, and similarly Python is also can be installed in your computer. And you can also use an excellent platform provided by Google, called Google Colab, which you will learn more as you go through the program. So this also deals with how to install Python and how to use Python in different environment. And one of the environment is Google Colab, that as I mentioned. And we go to what are the different libraries in Python that is necessary for the data analysis, and a lot of very valuable video embedded into these lessons that I would definitely suggest you to take a look at. And we need packages such as pandas and numpy to work with the data that we have, so that we can perform the necessary machine learning and necessary all the mathematical functions on the data that we have, which is going to be structured data. And you go through a different function that numpy or pandas provide you to analyze the data, to take a look into data, just like R. you can print out the data just like R. You can calculate the length of the data, size of the data, dimensions of the data. So it's really complementary, and the choice is yours. So similar to R, we have our own practice. We have Python's own practice, loading data or practice code blocks here, where we give you an introduction to how to what different sections of these code blocks means that I actually told you. And this is again a similar coding environment where Python codes are given with the slight changes in the syntaxes that will expand your knowledge in really understanding what are the important portions of the syntax and how it relates with the running a code successfully. So this also follows the same: how to import a text file, how to import a CSV file, how to view the file, or how to print out the file. And we give you a couple of more biological data sets for you to try and load and try and peek into the data, how it is represented, and what are the different aspects of these structured data. So again, just like in R, we deal with what are the functions and how do we employ functions in our basic code structure, and why is it necessary to use function in our code structure. And one change from the R environment to Python environment is at the end of every lesson, we give you a code challenge. So that challenge will actually test your understanding of what is the understanding of how did you process the underlying logic of these codes. So I encourage you to go through those coding challenges, and if you encounter any difficulties, we are always here to help you out. But if you go through the lessons and follow the lessons and the codes, these coding challenges are going to be really helpful for you to expand your knowledge in applying your understanding to a similar data set, or a different tweaked data set, or a different kind of analysis that's been spoken to you in these lessons. And yeah, that completes our review of how data science is structured in three basic in three streams of an understanding in this program. I hand it over back to Ilaya for further.
Thank you.